Webinar Wednesday series. My name is Malia Forbes, a marketing coordinator for ALS North America, and I'll be facilitating the webinar today. Before we begin the presentation, I have a few helpful notes. If you are having technical issues with our webinar platform, you can send me a chat message in the bottom right-hand corner of your screen, and I will be able to see your question. You can also select the hand icon to notify me of your status, and I'll be able to assist you. All questions will be answered at the end of the presentation. Our presenter today is Adam Rope. Adam Rope is Director of Sales and Marketing at ALS BioScreen. ALS BioScreen offers a wide range of services in human clinical, analytical chemistry, microbiology, toxicology, and stability. Adam has a background in engineering from University of Arizona and has been with ALS since BioScreen's acquisition in February of 2019 and with BioScreen since 2009. He is experienced with support services and problem solving for FDA-regulated clients, including pharmaceutical, personal care, medical device, and biotech companies. Today, Adam will be discussing OTC test planning. Adam, thank you for taking the time to deliver this presentation. I will now turn it over to you.
Hello. So we'll just get started on this. So the point of this presentation was to go through some questions that we commonly get from clients about how to plan for the testing associated with the new OTC formulation. So before we get going, we have our standard disclaimer that this is just from my experience and knowledge. It's everything in this, there is going to be some variance. None of the timelines are exact. It's just supposed to be a guide as to help people in terms of planning. So, and then at the end we'll go through some different timeline schemes that'll either get you faster or slower to market, but based on risk tolerance with cost associated. First off, why do we do any testing? Of course, it is a requirement of the FDA. I'm just going to refer everybody to the FDA's website in terms of any of the regulations on that. I don't really want to get into any of that here today. So the main questions we'll be answering, of course, is what testing is typically needed, what tests need to be done when, and what do you need done before you sell the product, or where should you be, and when is the best time to execute those tests, depending on how much, what the budget is, and how you want to allocate it. Oh, before we move forward, part of deciding about test needs, there are a number of different decision trees that can be employed. Here is one that we put together for deciding whether or not preservative challenge testing will be needed. Any water-based or water-containing formula, generally above 1%, will need a preservative effectiveness test. So if you aren't sure if your product contains water, or if you're pretty sure it's anhydrous, it's still a good idea to do a water activity and confirm that. It's much less expensive than AET, so if you think it's going to qualify as anhydrous, it's a good way to go first. So the first stage is, of course, your R&D tests. This is going to be that preservative effectiveness if you've got water in your product, any formula stability in glass, and your safety testing. So that'll be your 50 subject patch, any ocular testing, any tox reviews for products that can't be patch tested, and possibly elemental impurities if you think that might be an issue. We do have links on here to our webinar that we previously did on in vitro tests, and that goes through all of the different tests that we offer in that area, as well as where the technology is and where it's going, what's being researched, and as well as the link to the FDA's website on potential issues with elemental impurities, heavy metals in cosmetic products. So that second round with OTC products is you'll get into your efficacy tests. So for sunscreens and hand sanitizers, which I'm focused on today, uh, you would do your SPF testing or your kill studies for hand sanitizer. Um, and then you would do your method validation, assuming everything's looking good and you'll be moving forward with that product. And then you would be looking at doing in-package GMP stability, your drug stability studies. And we'll get into what is involved in each of these. So for SPF testing, you'll have this sort of gets done in two stages. You have your initial 3 subjects for the UVB testing that we always deliver first, just to make sure that you're, meeting the claim that you're expecting. If it's an SPF 30, that you're hitting that, or if you need to reformulate. That way, you're not out the cost of 10 subjects if you need to reformulate to get a higher claim. At that point, usually after 3 subjects, you can make a decision and move forward with your broad spectrum, and also start getting your stability going if you're happy with where things are at. So if everything looks good there and you move forward to the next stage, however, so that was for a sunscreen. We right now, due to a high amount of testing, we also wanted to include some information about hand sanitizers. The efficacy testing for hand sanitizers just was updated. Last year, the FDA put out the new proposed monograph, and it became official in April. The new test requirements are an in vitro log reduction, as well as an in vivo on hands glove juice clinical study. We are offering the log reduction kill test, and this is the list of organisms that the FDA is requesting. And then the glove juice test, unfortunately, at this time is not available um, due to the COVID-19 situation. Assuming that everything looks good in your efficacy tests, you'll move forward to analytical method validation. I wanted to cover a few important topics on this that we run across. One of the things that companies come to us asking for is a method that's already validated. Validation is formula specific. You We have methods that have been validated numerous times for lots and lots of different sunscreens, but when someone hands us a new sunscreen, the method has not yet been validated for that product. Uh, it will need its own validation, and validations need to consist of all of the parts of a typical USP validation. So every single sunscreen, it's a different drug, and that's how the FDA sees it. Anytime a formula is changed, there needs to be a revalidation. There's really very few corners to cut um, in terms of if you have a number of different sunscreens that have the same base and may have minor differences, there might be some ability to perform those validations side by side and save some economy of scale. And we can work with you to do that. But every formula should always have a full method validation. Following that, you would start your drug stability studies. Usually you'll start 3 lots that first year, and look for 1 to 3 months of data, depending on how confident you are in that formulation before you start selling the product. What we need in order to get your stability up and running would be a certificate of analysis that tells us all the tests and specifications. Um, the final components as the samples, it really should be in the type of packaging you're going to use, so that you can check for any sort of package compatibility issues at the same time. And of course, those first 3 lots of samples. We get reports out at each interval about 15 to 20 days after the pull point. We also did a full webinar on stability recently, and I threw the link on there for you if anyone wants to look at that. Some of the common release tests, which would also be part of your stability study, would be any of the physical tests, specific gravity, weight loss, pH, whatever you want to look at in that regard, as well as the micro, your typical USP 61, 62, and now that new chapter, USP 60 for Burkhold area cepacia. Um, and then, of course, you'll be doing that active ingredient analysis on every lot. So now we can get into some of the ways to stage your testing. So in that first set of R&D tests, uh, you know, a typical way you can go ahead and run them all at the same time if you have a high tolerance for any risk associated with any of the samples or formulas um, falling out. Um, Otherwise, our recommendation would be to start with preservative effectiveness and stability and then do the patch test later once it looks like things are looking good. Then you could get into your second set of tests like your efficacy, the SPF. You have that 3 to 5 weeks at first here where you get your first 3 subjects. If everything looks good there, then you can be off and running with your UVA, your method validation, and looking at your package compatibility. This is sort of a more conservative timeline that tries to save on budget in case a formula doesn't fare well. You can do a little more aggressive budget planning where you stack up the testing a little more. Of course, when you start tests and something might fail, you could be looking at more costs associated with that. But we did put a timeline together here that, theoretically gets you to that point in that in-package stability where you can have that first 4 weeks of data in under 20 weeks and get that sunscreen to market. Um, and we also put a hand sanitizer timeline on here as well, and you can see that it starts off with with those same tests, and then looks to get that formula stability done in about that first set of in-package stability done in at about week 14, week 15. In terms of getting any of these things up and running and planning things out, I would suggest that people engage their lab early. Um, we get a lot of calls when people are already in a rush and timelines can't really be hit. And I think it's important that you always let us know uh, what exactly is your reporting date that you need. What are your target dates? Is there an air date on, say, QVC or HSN that's being planned? Anything along those lines in terms of a launch date, it really helps, and we can give you options as far as how to progress and get things done to meet your goal. We can also try to pre-schedule testing so that it's on our schedule even ahead of samples being here. We just need to make sure that the samples can be at bioscreen in time for them to get started as planned. If not, there can be some cancellation fees with that. Uh, but we have our full customer service and sales team here to help, and I'm always available to try and answer questions and um, work through things and figure out how we get you where you need to be. So that brings us to the end of our presentation. So I think we can field some questions if there are any.
Thank you, Adam. We will open this up now for questions. You can type out your question in the chat section at the bottom right, and I'll give everyone a few moments to do that. You can also raise your hand, and I may be able to turn on your mic, and you can ask Adam your question. This webinar presentation is being recorded. All participants of the webinar will receive a follow-up email once the recording is available. We will also post our webinars on our ALS Global website and our ALS YouTube channel. I encourage you to find a wide range of video resources there and past Webinar Wednesday presentations. Also, please follow our showcase page on LinkedIn as we will post announcements and registration links for future webinars and other valuable resources and updates. Give everybody just a few more minutes. Adam, it looks like you did a very thorough presentation. Okay, so we have one question in the chat box. Which tests are needed for a hair care product? HRIPT, ocular, etc.
Yeah. Yeah. So two of those tests are correct. You'd also want to do stability testing for that. You would not need a validation of any kind for because you won't have any sort of an assay unless it's an OTC hair care product like an anti-dandruff product that has an active in it. Um, but it would be much more simple than a drug product.
Perfect. Um, we have another question. Would superficial wound care change the testing?
Yes, I think the efficacy testing would change. It also depends on uh, what if any monograph it falls under. There's a skin protectant monograph that some of those products fall under, and there's also a wound care um, monograph, and so it depends on whether it's an active or whether you're making cosmetic claims as opposed to drug claims and falling into that OTC category. But we would be able to take a look at that and figure that out with you.
Perfect. We have another question. Can you provide more detail on full
Yes.
analytical method validation? What steps are done? Do you need the customer to provide a placebo?
Yeah. So we can go back to slide 10. Um, so yes, we do need placebo. We also would need raw material of the active that's used. So if it's avobenzone, we would need your raw material to use as the standard for that validation. Um, we can also purchase standard if you wanted us to purchase like a USP standard or something from an alternative standard. But it's always best to use the raw material from the product. The stages are all outlined here in terms of what we go through in that process. We need what we need from the client is the formulation with the specifications, the of course the finished sample formula, the placebo, and that raw material, and then we would put together a the protocol and send it out for sign off. Once the protocol is signed off, we can move forward um, and go through each of these steps in here in terms of linearity and range, system precision, accuracy and recovery, specificity, robustness, any sort of those parts. So I'm going to answer the question 2 down before I go back, before I go to that elemental impurities question. But there's a question about LOD and LOQ. If you're, that would be more typical with something where you are doing trace analysis. So we would do LOD and LOQ if it's a validation for, say, trace heavy metals analysis in a drug product. But for something at higher levels, it wouldn't be typical. Um, so is elemental impurities required for stability testing? I would, we don't typically put the elemental impurities testing in the stability study. Usually it's just done um, on some initial either R&D batches or on a couple of the first production batches, or you could do it on any raw materials that you might suspect could be an issue. But it doesn't really need to be part of a stability study. What are the test methods? Okay, so one of the questions here is about what is our method? What are the methods for the hand sanitizers? So the methods that we're using are ASTM methods. I don't know I can't recall the number off the top of my head. Um, I but I would be able to send that to you afterwards. Both of these, the in vivo and in vitro, are both ASTM methods. Yeah. So there's a question about accelerated stability and going out to 8 months. No, we are recommending that those studies run for if you're making a claim of 1, 2, or 3 years. 3 months equals a year. So generally, the study runs for a full 9 months if you're making a 3-year claim, but a lot of clients start making decisions about moving to market and going into actual production after 3 months because many of our clients can't just um, wait another 9 months after they've already spent 4 or 5 months doing testing up until that point before they go to market. Vicky, I would have to get back to you on that last question. Uh, um, so we'll have to take that offline.
Adam,
So,
there was a question, is analytical validation required for antibac hand soap or only for sunscreen?
okay. So it's for any OTC with an active, method validation is required. If there's a claim being associated with an active, say you're making a chlorosilanol hand soap, yes, a method validation should be performed for the chlorosilanol assay for that hand soap. Time kill studies on hand sanitizers. The ASTM method, we would test all of the organisms that are on that list in on slide 9. However, we offer a more like an in-house method that is, I believe, 4 organisms or 5 organisms that we can do if you're just trying to range find on a hand sanitizer. And I do list a 4 to 5 week turnaround. It's really turning out to be more like 8 weeks actually for the log reduction on hand sanitizers. That is a typo that I just noticed. Um, do you agree that 4 months is relevant for stability by 40? I'm not entirely understanding the question. Um, Jorgen, if you can clarify your question, that would be helpful. Yeah, so once you have that 3 month data, Vicky, you'd be clear to do a 1 year shelf life. So it would be, once you get 6 months, it's two, you could do 2 years, and then 9 months is 3 years. Now, there's no direct regulation on this. It's sort of a rule of thumb slash, uh, industry practice, but that's what we would generally recommend is following that pathway.
Jorgen did clarify, he's clarifying 3 months times 4 equals 12 months.
Oh, so if you wanted to make a 4 year shelf life, yeah. So if you wanted to run your study out to a 4 year claim, which isn't very common for us to see, yeah, you would just add another 3 months on.
So for the hand sanitizers, is MIC required? Can any data from AET be used as part of the efficacy testing?
So minimum inhibitory concentration, you typically do that on the raw material that's being used for the kill. So that would be on the alcohol or if there's another active that's being used. You wouldn't really do that on the finished product. So um, we don't get asked for that. Typically, um, hope your raw material supplier would have that information. If not, yes, FDA does have it in their monograph, um, and it would be on the same set of organisms. Uh, AET is completely different from log reduction, and it's not particularly applicable for alcohol-based hand sanitizers in particular. I wouldn't say that AET could be used for in place of the log reduction testing at all.
Okay, what is your method for testing alcohol
Yeah, for testing.
concentration for stability?
Yeah, in that case, it's a gas chromatography method. It's an in-house assay, but we're doing that all the time right now. So we have that ready to go if you're looking for that.
Okay, we have a question for antibac hand
Yeah.
soap. Can R&D lab tech, I'm sorry, can R&D lab batch be used for all required testing, stab, efficacy, etc., or is that required to be done with a production batch?
So yeah, you can use a lab batch for pretty much for all of your R&D testing. We would just recommend that when you go to do your GMP stability that you're using a production batch.
With the PEG, I'm thinking of the 1,4-dioxane claims. Your thoughts?
Um, yeah, no, we can test for 1,4-dioxane if you're looking for it. How can you keep testing costs low? The best ways are to put a complete package together for for all of the associated testing, and plan it out as with the same lab. The other option is to stretch things out more in case anything fails along the way, that you do things one step at a time rather than trying to move quickly. The choices are really uh, taking more time and being lower risk with budget, or trying to move quickly and tolerating a little more risk. But we can assist you in trying to figure out what the best path for your budget is. I'm making an assumption, Malia, that other people can see the questions that I'm reading.
No, not until. So this question, are you looking at this one? There is no direct regulation on the stability duration requirement for cosmetics. Is this the case also for an OTC?
Okay. Is there no duration on stability duration? So that is correct. There's not really any direct regulation on stability for cosmetics, other than, if you're making a claim, you should have data to back it up.
We have a
But
question. What
OTCs
kind
are
of,
I'm
sorry.
sorry, but I'm sorry that question. My answer was somewhat incomplete. In the case of OTCs, there is regulation because OTCs are not cosmetics, they are drug products, so their regulation falls under drugs, and stability is regulated for OTC products. How can you keep test cost low for SPF products? So this question was about keeping testing costs low for SPF products. That's a difficult area to do that, depending on what and low is somewhat subjective. Um, there's a lot of testing involved with sunscreens. There aren't any shortcuts to take. My best advice would be, if you're concerned about cost, to do things more stepwise and take more time. What kind of test for residual solvents? So the last question here is about residual solvents testing. We do offer that. We use the USP 467. We haven't done that on formulations of this kind of complexity uh, for sunscreens. It's pretty difficult to test for those. Um, we see that more in pharmaceutical drug products, which have many less excipients than a sunscreen would.
Perfect. It looks like that's all of our questions for today. We thank everybody for attending. Thanks, Adam, for giving this presentation and taking the time to answer everyone's questions. We hope everyone enjoys the rest of your week and look forward to the posting of this webinar on our YouTube channel.